So we talked about the nucleus structure, right? So it has the nuclear envelope, okay? So membrane is double membrane and nucleus, and also it had a condensed region, which is called nucleolus, okay? So inside here, the rRNA, which is the ribosomal component, is synthesized, okay? So this was the rough ER, right? So after the RNA was synthesized, this gets translated into the protein, okay? So protein synthesis actually occurs in the ribosome that is stuck to the surface of the rough ER, okay? So that enters the inside of the rough ER all the way to Golgi and then to the plasma membrane throughout the vesicles, okay? So as this goes through rough ER and Golgi body, the modification of protein actually occurs. And then these were the chloroplast and mitochondria. This was the organelles for cellular respiration. And then we also talked about the cytoskeleton, right? So microtubule is like a hollow tube, which is very thick, okay? So its diameter is almost 25 nanometer. So compared to microfilament, its diameter is much bigger, okay? So it had special functions for microtubules, okay? So other than just shaping the structure, it also forms spindle fibers during the cell division. Also, it can form cilia and flagella. Okay, so this is a structure this is the cross-section of cilia and flagella. So cilia and flagella actually has 9+2 structure where 9 pairs around and then 2 microtubules in the center as well. Okay, so this is a flagella and cilia structure, and this moves back and forth like this to actually sweep something out, okay, or to actually make the cell mobile. And we also talked about the microfilament. So one example of microfilament in the cell is the contractile ring. Okay, this only appears in the animal cell. All right. So after the DNA was duplicated and then divided, the cytoplasm also has to divide. Okay. So in that process, the contractile ring actually gets formed. Okay, and that's made up of microfilaments. Okay. So that actually tightens up. Okay, and lets the two cells actually divide like this. And then another example of microfilament in our body is the muscle cell, right? So muscle fiber actually has these kind of like smaller fibers inside, okay? So these were all microfilaments actually stacked together, okay? And then after that, we talked about the membrane structure, right? So on the phospholipid bilayer, other than the lipid molecules, we also have these proteins stuck, okay? So penetrating through the membrane, we have these kind of structures, okay? So out of those, especially, we focused on the transporters, okay? And these were the tertiary structures. So briefly, we talked about these kind of transports, okay, in and out of the cell, right? So we talked about the simple diffusion like this, which is a part of the passive transport, okay? So this just goes through the concentration gradient from high to low. This also is a passive transport, but this is the facilitated diffusion, right? So through the channel that's specific for this solute, okay? Actually, this channel opens for this solute only, okay? And then this goes through the concentration gradient again. Active transport is kind of different from these two. So active transport actually uses ATP as the energy to pump out something, okay? So from low concentration to high concentration against the concentration gradient, the solute gets pumped out, okay? Also, for another example of active transport, we talked about endocytosis and exocytosis, okay? Endocytosis, we talked about 3. We talked about pinocytosis, phagocytosis, and receptor-mediated endocytosis, okay? So these were all taking in the molecules to the inside of a cell like this, forming a vesicle, and exocytosis is the other way around, like this. Okay, and then we looked at the channels. This is the facilitated diffusion. This is the pump, okay, active transport. And this was another example of pump, okay? So this is sodium-potassium pump. So note that the pump can co-transport, okay? So it can let out sodium and let in potassium, okay, against the both concentration gradients. Also, this was the phagocytosis. So this was another type of endocytosis, do you remember? Okay, so here pseudopods actually gets formed. Okay, remember the pseudopods is a microfilament, right? Okay, so after actually it goes through phagocytosis, okay, this cell is a white blood cell. Okay, so this is like immune cells that actually fight for the bacteria or foreign molecules. So if this big bacteria actually gets taken up by this white blood cells, then lysosome actually gets fused with this food molecule. Okay.